And we're live.
All right. Well, welcome back, everybody, for the pièce de résistance of the day, keynote lecture by Dr. Eve Marder. It's our very profound pleasure to welcome her to this fourth edition of our conference. Eve Marder, as probably doesn't need any introduction. She's, a seminal pillar in our field. But I'll read a few things here. So Eve Marder is the Victor and Gwendolyn Bindfield Professor of Neuroscience and Biology at Brandeis University and member of the National Academy of Sciences. Her work focuses on the dynamics of small neural networks and was instrumental in demonstrating that neuronal circuits are not hardwired, but can be reconfigured by neuromodulatory neurons and substances to produce a variety of outputs. She employs electrophysiological, biophysical, computational, anatomical, biochemical, and molecular techniques, so the whole gambit. Eve has received many honors from her pioneering work in the field, including election to the National Academy of Sciences, the Miriam Salpeter Memorial Award for Women in Neuroscience, and the George A. Miller Award for the Cognitive Neuroscience Society. And in 2016, the very prestigious Kavli Prize in Neuroscience. And just before we got live here, we were just discussing in the green room, and Eve said that had she known that all the wonderful talks that we heard today, we were treating some of these questions, she would have added slides. Apparently, she has answers for all the outstanding questions that were left unanswered, but hopefully she can enlighten us on other questions. So today, Dr. Marder is going to talk to us about differential resilience to perturbations of circuits with similar performance. And with that, I leave you the floor.
Oh, thank you very much. I'd like to thank the organizers for giving me this opportunity to speak to such a diverse and highfalutin crowd. I should say that I hope all of you realize that I am a card-carrying neuroscientist and I'm a reductionist neuroscientist. So I feel that I have many things to say to people who worry about brains and consciousness, etc. But I'm going to come back down to basic principles about how neurons in real brains work. And I have all sorts of things that I'm not putting in today's talk, but we could possibly talk about them later. So, but what I do want to talk about is a topic which sort of came up in several talks already today, which is the differential resilience to perturbations of circuits with similar performance. And this deals with the whole problem of animal variability or animal-to-animal variability, human-to-human variability, and what all the fundamental biological questions that get raised by the variability in our populations. Before I get going, I would just like to thank my present lab, the postdocs, some of whom have given material to this talk, my PhD students and others. And then just again to acknowledge all the people with whom I've had the privilege to work. And I like to show this slide before talking about data because I really deeply feel that everything interesting I've ever done is because one or other of these people really drove us in new and different directions. And for all the young PIs out there, I would just like to say that you should listen to your people. To all the young people out there, just remember the future lies ahead, and there are, all sorts of wonderful futures in science, and just keep going. Okay, so the problem. How is individual variability related to resilience to challenge? And this is sort of a very, very deep question in all of biology. How do biological systems, which differ from individual to individual, whether it's individual cells, individual organisms, how is their variability related to resilience to challenge? And of course, it's become very, very acute in today's world as we wonder why, of all the people who get infected with COVID, why some people become very, very, very sick, why some people die, why other people don't. And this all goes back to what are the fundamental biological mechanisms that determine resilience at every level of organization. Now, I I've spent my entire scientific career working on what used to be called a model system. I don't like that term. I call it a small nervous system. This nervous system is part of the stomatogastric ganglion of crustaceans. In particular, we work in lobsters and crabs. And I've worked in this nervous system for my entire career for several very important reasons. Reason number one is that this nervous system, when dissected from the animal, and I'll describe to you how we do this, continues to generate motor patterns which can be recorded in the dish that resemble almost perfectly the motor patterns that you see in the animal. And the reason I care so deeply is if you want to understand how to account for a circuit's behavior in terms of its underlying cellular mechanisms, then it's really important to have a circuit whose behavior can be recognized as normal or not normal, or is changed or altered, or is modulated, and that can be very easily measured. So to go back to the beginning, what do? Well, in the pre-COVID days, we would call up the fish market distributor on the waterfront at Brandeis. We're about 10 miles west of the ocean. And they would put the animals in a taxi, and the taxi would come to campus. In the COVID days, we're not allowed to have taxis bring animals to campus. So my wonderful technician goes down to the waterfront and picks up the animals and brings them back. Then they go into seawater holding tanks. These are Cancer borealis, the animals we're working on right now. That's the crab. It's about 5 or 6, oh, you can't see my hands, about 5 or 6 inches large, and they're 5 to 7 years old. And this is important. That means these animals have lived in the North Atlantic Ocean for 5 to 7 years before we get them, which tells you that they are actually capable of dealing with a real life environment. So we hold the animals for a while, then we bring the animals up to the lab. We take the stomach out of the animal. We dissect the stomatogastric nervous system off of the surface of the stomach and pin it into a Silgard line petri dish. This is a few inches across. I don't know, it's a regular sized petri dish. It's probably 5 or 6 inches across, maybe a little smaller than that, maybe 5 or 6 centimeters across. And what you see here is the nervous system as we usually record it. In the center is the stomatogastric ganglion. It has, in crabs, about 26 somata, and they're very large. They're about 100 microns. So that means it's easy for us to put 3, 4, 5 intracellular sharp electrodes into the cells to record intracellularly. And that gives the kinds of recordings you see here. We also record extracellularly from the motor nerves. This is a motor system, and this ganglion used to innervate all the 40 sets of striated muscles that move parts of the crab stomach. And I should say a crab stomach is nothing like your stomach. A crab stomach is much more like your respiratory center, or like your jaw, or like your shoulder. It's a locomotory system with excitatory motor neurons that drive muscles in a repeating motor pattern. Now, what those recordings, so the other important thing for me to tell you is we identify the neurons unambiguously on the basis of what muscles they used to innervate. And we do that by putting as many as 20 pairs of extracellular recordings on the motor nerves. So this preparation has generated 50 years of simultaneous recordings from every neuron in this circuit for miles and miles and hours and hours and hours and hours and hours. And we're still finding really interesting questions with which this preparation can be useful for. So when people tell me that they have X number of hours of simultaneous recordings from 3 neurons, I start laughing, because it's very, very interesting how difficult it can be to extract all the information from multiple sets of simultaneous recordings. But anyway, what do those recordings look like? This extracellular recording, down here you can see activity in a neuron we call the LP, PY, PD, LP, PY, PD. The pyloric rhythm is a triphasic rhythm, as you can see. The LP is a constrictor motor neuron, or it's part of the constriction of a valve at the back end of the stomach. The PY is another constrictor, so this is constrictor 1, constrictor 2, dilator. That's the dilator for for this valve at the back end of the stomach. So we have constriction 1, constrictor 2, dilator, constrictor 1, constriction 2, dilator. And these intracellular recordings are showing the membrane potential waveforms that give rise to this motor pattern. And you can see here, these cells depolarize, they fire a burst of action potentials, they hyperpolarize, etc. And here you can see what in 1980 was the Holy Grail. We then called it a wiring diagram or a connectivity diagram. Today, this would be called a connectome. And in the late 70s, the goal was really to figure out what the wiring diagram was, because the assumption was that once you had the wiring diagram or the connectome, you would understand how this circuit worked. And in 1980, when we and people in other preparations had these connectivity diagrams, we realized that the connectivity diagram was the beginning and only the beginning of understanding how neuronal dynamics come from this wiring diagram. And this, of course, is as true about the connectome in vertebrate preparations today. What I would like to say is that a connectome is absolutely necessary and completely insufficient to understand neuronal dynamics or how a circuit works. In this particular connectome, and any others I might show you, this resistor symbol means electrical coupling, and these filled circles are chemical inhibitory synapses. So the first thing you'll see in this circuit is that there's no excitation. There's no fast excitatory synapses. The way this circuit works is that the AB, which is an interneuron, is an endogenous oscillator. It's electrically coupled to the PD neurons. Therefore, the PD neurons fire in bursts. Together, these two neuron types inhibit the LP and the PY. The LP neuron recovers from that inhibition first, and it holds down the PY. Eventually, the PY recovers from its inhibition and shuts off the LP, and then the circuit, the whole cycle repeats itself. Now, I think it's clear that this connectivity diagram is missing all the information that would allow you to predict these dynamics. And indeed, if I just showed you this connectivity diagram, there'd be no way you would be able to know what these dynamics are. And that's because what's missing in all of these simplified diagrams are the identity of all the voltage and time-dependent currents and the strengths and time courses of all these synapses that would allow you to understand how this rhythm arises from the essential properties of the neurons and their connections. So what we and many others have been doing since 1980 is trying to go from a simplified word model that I just gave you to understanding how these dynamics arise. And to do that, we started out doing a lot of biophysics and measuring all sorts of currents. And then we realized, and I think it's important for this audience to know that by 1990, it became completely clear to me that even if you had all of the biophysical data describing all the individual properties of all the synapses and all the cells, you would still need some way of putting all that information back together again. And that's why we started building computational models. So it was the reductionist biophysicist in me that drove me back to having to build models in order to understand how anything worked. So moving on, I'm going to show you the kinds of models we most usually use. Now, these model neurons are single compartments. They usually have about 8 conductances. So here's a round single neuron, and they routinely have an inward sodium current. They routinely have 2 different calcium currents. They routinely have a hyperpolarization activated inward current, or IH. This is a current that's very important in many oscillators, including in your hearts. And then they have 3 potassium channels. This is one called the delayed rectifier. That's the one that you learned about when you studied Hodgkin-Huxley, those of you who took a neuroscience course. And then 2 others, a calcium-activated potassium current and an A current. That's a transient fast outward current. Now, this is a whole lot more complicated than those of you who are used to looking at rate models and neuronal models of that sort, and a whole lot simpler than what a real neuron is doing. But for us, it's a good heuristic because it allows us to ask some pretty specific sets of questions about how neurons work and how the circuits work. So before going further, I would like to show you, for me, one of the most wonderful visualization tools that Leandro Alonso, who is a postdoc in the lab, developed recently. And if you think about it, if you want to understand how a cell, how a cell changes its membrane potential, this is a bursting neuron. So it's going along, it starts depolarizing, fires a burst of action potentials, and then comes back down. What you really want to understand are the sequence of the activation and inactivation, the turning on and the turning off of all of the voltage-dependent currents in the cell. And you'd like to know what's happening at all times, but we had no particular way of really seeing all that happening at the same time. So Leandro developed this visualization method, and in this, in the middle, we have a line here, and the outward currents are plotted up, and the inward currents are plotted down. That's using the convention that biophysics usually uses. The total outward current is seen here on a compressed log scale. So you can see the outward current gets much bigger here. The total inward current is seen down here, again on that. And now we can see the outward currents now as a percentage of the total outward current. So you can see, for example, in purple is the A current. It starts out really small and it gets bigger. In orange, you see the calcium activated potassium current. And you can see it starts out reasonably small, but it gets to be humongous down here at the end of the burst. Here you can see the sodium current turning on and then on during the action potentials here. And you can see various other currents. So this allows us to see at a glance what all the different currents are doing, what roles they're playing. They're turning on and turning off in the dynamics. And I'm going to use this method to show you the first important lesson of my talk. And I think this came up in a way in one of the previous talks. And this is what I call multiple solutions or degenerate solutions producing similar behavior. First, we're going to see at the level of a single neuron. So what Leandro did is he used a series of genetic algorithms to find a whole bunch of neurons with very, very similar behavior. So this slide is looks more complicated than it is, but here's one bursting neuron, neuron A. Here it's bursting. Here's neuron B. It looks very similar. Neuron C. It looks very similar. Neuron D, E, and F. So you have 6 examples of bursting neurons, and their output patterns look very, very similar. Nonetheless, if you use Leandro's currentscapes, you can see, if we just look at the A current, this purple current, it's tiny here. And if you look down here in F, you can see it's enormous. If you look at, in orange, the calcium activated K, you can see it's pretty big here, but its role in the cycle is different in each one of these models. And if you just use your Gestalt viewer, you can see that these sets of currents, while similar in some ways, look actually quite different. And so, for example, here in brown, you see the H current, which is pretty big here, and it's completely missing here. This one doesn't have an H current. So the first thing this tells you is that you don't need the same set of conductance densities, or for that matter, necessarily even the same sets of currents to give very, very similar dynamics. And I would just like to make one more point, and that's to say, if we look here at the actual conductance densities, the number of A current channels in A was very small. But here, where the A current is much bigger in terms of the dynamics, it's actually a smaller number of channels in that membrane than, for example, in model C, where the A current itself is much smaller. So this tells you that the role of the current is related to, but not simply determined by the number of those channels in the membrane. Okay, so here we have the principle of multiple solutions. Now, this is a slide that many of you have probably seen before. This is work that Astrid Prince did, and it's the same lesson at the level of the circuit. So years ago, Astrid wanted to build a model of the pyloric rhythm, and so she ended up building 20 million, simulating 20 million model networks, and then selecting from those ones that were good models of the pyloric rhythm. And she ended up with 400,000 models that she considered good pyloric network models. And what this did for us, and this was back in 2003, 2004, it made it clear to us that the goal should never be to build a single model that was sort of intended to characterize the mean behavior of a lot of data. Instead, we now always build populations of models which are determined or designed to capture the behavior of a whole population of biological systems. So, but just let's look at these two circuits. Those two circuits are doing pretty much the same thing. But now if we look at their underlying parameters, you can see their underlying parameters are totally different. Just like you saw in the previous slide, at the level of single cell, here you're seeing it again. Now here are some of the synapses. In this network, the AB to LP synapse, that's one of those inhibitory synapses, is big. Here it's tiny. The PY to LP synapse in this one is big. Here it's small. The H current in the PY is big over here. It's small here. And you can say this set of parameters and this set of parameters are completely different, but nonetheless, both are capable of producing circuit behavior that looks very, very similar. So when I first saw these data, I loved them. And I loved them because it instantly freed me from the biologist's nightmare. And the biologist's nightmare is worrying about the variability you see in your data, worrying about, we were always worrying that all the variability was our fault. And what this immediately told me is that some amount of the variability, and possibly a lot of the variability that we would measure in real preparations, was actually biologically real, because we saw here the proof that you could get very similar behavior with differences in underlying properties. When I started talking about this, I got a tremendous amount of flack from theorists. And the important piece of the flack, which I actually listened to, I do sometimes listen, was they said, and rightfully so, that this set of parameters and this set of parameters, while consistent with producing a very similar behavior under these static conditions, there's no way that an animal that had these sets of properties would behave in response to perturbations the same as an animal with these set of parameters. And so I started thinking about this, and I said, it's true. The question is not whether there's a perturbation in the universe of all possible perturbations that will separate these two, because there has to be a perturbation that will separate them, because they're different. However, the question then became, if I took this seriously, I said, if this is really crab 1 and this is really crab 2, can those crabs respond reliably and reproducibly to the perturbations that those animals are going to see in their life? So this, and then it also raised the question of how big are the variability in the underlying data. So the last 15 years have been really trying to answer these questions. How variable are the parameters in real biological networks? Do perturbations reveal cryptic animal-to-animal differences? How reliably can animals with intrinsic variability respond to perturbations? And are some sets of parameters associated with higher resilience? So I'm going to briefly take you through, we more or less know the answers to these three. And this, I think, is, a really interesting question among others that we're still thinking about. So the first thing we did after we got those computational data was to go back to the animals and measure the variability in some of the properties we could. So there's one LP cell in each animal. So this is LP from crab 1, this is LP from crab 2. You can see in the network, their dynamics look very, very similar. But when Jean-Marc Gaillard went into voltage clamp and actually measured the amount of current, here's just the calcium activated K. You can see it's very big here and it's much smaller here. The A current, again, it's much bigger here than it is here. And across animals, and Jorge Goash had seen very similar things years before, we see basically a 2 to 6-fold range of measurements of the same current in the same identified cell across animals. And then when Dave Schultz took those same cells and he pulled them out and used single cell RT-PCR to measure the mRNA copy number in those same neurons, again, you see this 2 to 6, 2 to 7-fold range of expression of the genes that give rise to these currents. And interestingly enough, for some of these currents, there's a very strong correlation between the current densities we measure in voltage clamp and the mRNA we measure in PCR. Now, this was from the first set of experiments in 2006. This is 2020. In some ways, it gives you exactly the same answer, but it adds something that it's going to become very, very important. These are data that we've collected with Dave Schultz in his lab. And now we see 20 currents. These are 20 genes for ion channels that have been measured in single PD neurons. Okay, remember there are 2 PD neurons in each animal. So this is showing you the range of expression of all these channel genes in identified neurons across the population. Now, you might immediately ask yourself, why do we have so many different channel genes expressed in a single neuron in a crab's stomach that doesn't have to do very much? And I would just remind you that every neuron in your brain has between, call it, 15 and 30 different kinds of ion channels in its membrane. And one of the really interesting things we're starting to think about is what are the benefits of having so many different kinds of ion channels in the membrane of a given neuron. And I'm going to come back to that at the end of my talk. So the first thing I want you to see here is a single neuron. These are identified neurons. They're unambiguously identified. Each channel gene shows that 2 to 6, 2 to 7-fold range of expression across animals. And interestingly enough, I think you'll be relieved to know that the channels we measured in 2005, 2006 are the ones that are big in these data. And some of these little ones are ones that we didn't know were there until much later. But single neuron has 20 different kinds of ion channels, maybe more. But it's of order 20. It's not 2. It's not 3. And it's not 5,000. It's of this order, and it's a number that I think you should think about. And I also want to remind you about this 2 to 5, 2 to 6-fold range of parameters, because that's the range we and everybody else gets whenever you measure any identified parameter, whether it's in mouse or in lobster or in leech, you get this back out. And I think that's, again, it's a magic number. Okay, we'll come back to that. The other thing we found in the early work is that there were some very strong two-dimensional correlations in the expression. One of the most dominant ones is a very strong correlation between the expression of the HCN gene, that's the one correlate for IH, and shell, that's IA. And we find that in many different cell types. But the other thing we discovered is that the correlations that we measured among these different channel genes, which were found in all of the cell types, were cell type specific. So every cell has many channels. There's a cell-to-cell variability, but there's a different set of correlations that in each cell type, and we're going to come back to that later again, hopefully. Now, with that variability, I'm just going to show you just some extracellular recordings, and these were 5 recordings from 5 animals that Adriana Topolik made just for this point. This is animal 1, animal 2, animal 3, animal 4, animal 5. You can see the pyloric rhythm, LP, PY, PD, looks very, very similar across those animals, despite the fact that if we were to go in and measure any of the underlying parameters, we would recover that 2 to 6-fold range. And here you see the same thing with the intracellular recordings. This is PD, animal 1, animal 2, animal 3, and you can see these waveforms are very, very similar. And this is the LP, and this is the PY. So this tells you that despite that 2 to 6-fold range, the intracellular waveforms are maintained as well as the extracellular motor patterns. So you've got to be thinking about what kinds of correlations are there that will allow these very, very robust and similar behaviors to be expressed. Okay, so just summarizing that, individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when they have similar behavior. Now, this is as true in the human brain as it's going to be in a crab brain. And I think one of the important things we have to think about, and I think we can discuss this later maybe, is why this 2 to 6-fold is so important. Okay, now, the next thing we wanted to do is we wanted to say, okay, if we have this variability, what happens to perturbations? Because again, those pesky theorists had said that animals with these different underlying sets of parameters would respond differently to perturbations. So we were looking for perturbations that would be natural, ecological for the animal. And obviously, the most obvious one is temperature. If you work on a cold-blooded animal, such as a crab that lives in the Atlantic Ocean, the water temperature changes for our animals in their habitat from about 2 degrees to about 25 degrees normally. And within a day, it can vary 10 degrees depending on, these animals tend to be intertidal to go down to about 600 feet. So depending on how deep they are and what the tides are and what the currents are and what the weather is, they can see a 10-degree change in temperature within 24 hours. So the reason why temperature is so interesting is because for those biologists among you, you remember that temperature is a global perturbation that influences every biological process, but does so differentially so that the relationships among the processes will be altered. We've also studied pH, because again, pH changes as water temperature changes, and we've studied high potassium, and there are other perturbations we're thinking of studying as well, but most of what I'm going to show you now is about temperature. Now, biologists are taught in college, as I was, that a Q10 is a characterization of how much a process changes as a function of a 10-degree centigrade change. So if you have the process here, a Q10 of 1 means it's temperature invariant, a Q10 of 2, which is shown here, a Q10 of 3 is shown here. Now, most biological proteins have Q10s in the 1.5 to 4 or 5 range. Most ion channels have Q10s for the activation and inactivation, the rate constants of their turning on and turning off in the 1.5 to 3 or 4 fold, except for the Q10s of the molecules that are important for temperature sensing. So we have specialized proteins which are important for sensing temperature, and they can have Q10s up at 20 or 40 or 60. But those are not the ones that we're going to be talking about today. Today we're going to be talking about the molecules that are important for the dynamics of neuronal signaling normally, and they all have Q10s in the sort of 1.5 to 4 or 5 fold range. Now, if you think about it, if you do the thought experiment, and that's what I want you to do now, you'll realize that if every single process that gave rise to neuronal dynamics, the opening and closing of sodium channels and potassium channels, etc., if they all had the same Q10, then the behavior could be maintained. However, if they have different Q10s, you can easily imagine all those processes will get out of whack as you start changing temperature, because that specific timing cues will get changed. And that's shown in this slide that Tim O'Leary made for a paper about temperature, that this is a model. It has the same set of starting conductances, but Tim has given 3 different sets of Q10s to the 3 versions of the model. So if you start at 10 degrees, in green here, you can see 3 versions of the model with the same behavior. one set of Q10s, when you warm up the model in the computer, it goes from being a beautiful burster to being a single spike burster, and then it depolarizes and crashes. Whoops. Q10 set number 3 goes into a single spike bursting mode when you chill it down. This model, Q10 set 2, is the most well-behaved of them, and then it goes slower as you go down in temperature. It speeds up for a while, and then up here, it starts firing tonically. But not single one of them maintains its behavior as a function of temperature. And if you just search in random parameter space in neuronal models, it's very difficult to find a robust model. And this will become important in a minute. However, what do the animals do? What do my crabs out there in the ocean do? So these are old data that Lamont Tang collected a number of years ago. He's now recording extracellularly the pyloric rhythm. And here you see PD, LP, PY at 7 degrees. Here you see it when he raised the temperature to 19 degrees. And you can see a beautiful triphasic rhythm at 19, a beautiful triphasic rhythm at 7. And he sees, if you plot the frequency of the network as a function of temperature, you get basically a Q10 of a little bit over 2, which is perfectly normal. Temperature makes it go faster. But what completely blew my mind when Lamont brought these data to me is that the phase relationships, that is to say the normalized time in the cycle that each cell starts and stops firing, is completely temperature conserved. So here you see the PD on is the reference point, PD off is a function of temperature, LP on is a function of temperature, LP off, PY on. So the cycle, LP, PY, PD, LP, PY, PD, that set of phase relationships, which is what's important for the motor system to work, for the timed activation of each of the muscles, that's perfectly maintained over this fairly large range in temperature. So biology has figured out, despite the fact that it's variable, it's found solutions that allow it to be phase constant. Now, however, and this just shows you the same thing with intracellular recordings. It's just a pretty slide. 7, 11, 15, 19, 23. Here, Lamont just superimposed the waveforms at 7, 11, and 19. You can see how beautiful the waveforms are. So despite the fact that the channels are opening and closing with different Q10s, and they do have different Q10s, there have been compensations so that the phase relationships are maintained. And that's now you're seeing up at 23 degrees. So you're seeing things from 7 to 23. What happens when you go to more extreme temperatures? So Lamont did this, and in the year that he did it, he discovered when he went to temperatures warmer than 23 or 25, he saw very different kinds of what he called crashes. These are recordings of 4 different animals, 1, 2, 3, 4, and they all are showing disruptions or crashes that were characteristically different. And if you think about it, this is exactly what you would expect if each animal had a different set of underlying parameters, and then they could all handle the temperatures that those animals actually had evolved to handle. But then when you went to a much more extreme temperatures, they would start showing the consequences of those underlying parameter differences. But, and here's the big but, and this is something I just want to show you. When Sarah Haddad, who was a beginning graduate student in this year, came to the lab, I just asked her to repeat Lamont's data, and she came back and she said, they're not crashing. And I said, surely they're crashing. I said, you just don't have the thermometer in the right place. And she went back, and a day later she said, they're not crashing. And she argued with me, and I said, oh, come on. And then she came back and she said, at 36 degrees, it's going 7 hertz. And my mouth dropped open because I'd never seen a pyloric rhythm faster than 3 hertz. So in the in the spring and summer of 2012. This was after the winter that the water never got cold. We have here 11, 20, 25, 30, 36. We had perfectly good pyloric rhythms at temperatures that Lamont would never have seen a good pyloric rhythm at. And this last summer, we also had very shifted high levels of temperature. So the crab habitat that winter, the winter before these data, was 6 degrees centigrade warmer than normal. In Woods Hole, they discovered that the squid had all gone to Maine, the dogfish had all gone to Maine, and we were recording crabs that were obviously acclimated or adapted to these very, very high temperatures, except we, of course, don't know whether we were seeing the population that was able to survive this very, very warm winter and all the others were dead, or whether all of our crabs had gone to Maine and we were seeing crabs from warmer locations. In any case, the next year, the weather got cold again and our crash temperatures went back down. But this is, so now, but I wanted to remember to tell you one more thing. Usually we only recorded 11 degrees until we started doing these kinds of perturbations. At 11 degrees, we never would have known that this animal was different than the other animals we would normally record at 11 degrees. So if you don't do perturbations to test the consequences of these extreme things, you would have these cryptic changes that will probably alter other things as well, but you never know if under your control conditions whether those changes are there. Okay, but when moving along, Leandro wanted to find models that would capture these behaviors. And so what he did is he used genetic algorithms to find pyloric networks that were temperature compensated, that here we have 10, 15, 20, 25. He wanted them to be like Lamont's data and to be phase constant all the way from 10 to 25 degrees, but then to crash at 30 degrees, which is what they do. Now, these are not easy to find, but using his genetic algorithms, he found them, and he found a whole bunch of them. So I'm going to show you a little bit of what you see. First of all, here are 5 models. And just like Lamont's, and these are at slow time basis, and these are expanded time basis. So if we just look here, animal 1, animal 2, animal 3, animal 4, animal 5, it's actually really model 1, model 2, model 3, model 4, model 5. You can see they're all showing different kinds of semi-crashes. And that's because they all actually have different underlying parameters. And we, of course, can go back and see what those parameters are and what their correlations are. And a lot of that is discussed in this paper that was just published. Okay, so the conclusions from the crashes were, at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation. Each individual animal crashes with individual dynamics as predicted by their underlying parameter differences. And then I didn't show you these data, but Sarah discovered that some neuromodulators can actually expand the stable range of temperatures, while others make the preparations more temperature sensitive. Now, I want to now show you my favorite slide of 2020, because this goes back to the issue about why you may have so many different channels and membranes. What Leandro did is he's just plotting here the last spike in a burst at 10, 11, 12, 13, all the way up to 25 degrees. So you're seeing the last spike in the burst from that bursting neuron. And we're using the same method I showed you before. So at 10 degrees, you can see in purple a very large component of A current. And as we go warmer and warmer and warmer, you can see that the A current is getting smaller and smaller. In orange, you're seeing the calcium activated K. It starts out small and it's getting bigger and bigger and bigger and bigger and bigger and as you go up to 25 degrees. Now, the last spike here can repolarize the cell just as count here, but you're seeing here a smooth trade-off in the job or the work done by these potassium channels in repolarization. Here you've got one set of solutions, and now here's another set of solutions, but you're not seeing any disruptions in the behavior. Instead, you're seeing a really smooth transition between the jobs or the roles of these different potassium channels. And I think that one of the reasons why you have so many different kinds of channels in cells is so that you can expand the dynamic range of neurons so that they can smoothly move through mechanisms, and they're not held hostage to the fact that each of their channels has fixed time constants. So these are all potassium channels, but they have different rates of activation and inactivation and different voltage dependencies of activation and inactivation, and the cell manages to do a very smooth trade-off in terms of those mechanisms, and that's seen here with the inward currents. If you look at the green, blue, and red, you can see again those ratios are changing, so the work done by those channels is smoothly changing in response to this perturbation. So we're expanding the dynamic range over which the cell can work. And one of the things we're starting to do now is to ask, what happens if you now add additional currents? Can we actually see that again? Okay, just a couple more slides from this. And this is again from Leandro's work, and it's just to reiterate the same thing. These are two different models. Now we're looking at 10 degrees and 25 degrees. Here we're plotting the sodium current, and it's small at 10 degrees, and it gets bigger at 25 degrees. In this other model, here's the sodium current at 10 degrees, and then 25 degrees, it's getting smaller. Likewise, if you look here at one of the calcium currents, it's small at 10 degrees, it gets much bigger at 25 degrees. In the other model, it's big at 10 degrees, it gets smaller at 25 degrees. So this tells you that even though both of these are perfectly good oscillators and both of them increase in oscillator frequency when you increase the temperature, that you don't necessarily expect that temperature will have affected any one of the underlying processes in the same way. And so again, this is one of the consequences of the underlying variability. Now, just one more of these for those of you who are pharmacologists at heart. This is now a network, PDL-PPY, and the A current, that's one of those potassium currents, is at 100%. And here Leandro has turned it off at 10 degrees, and you can see the network is completely disrupted. And now he does the same thing at 25 degrees. Here's the network at 25 degrees. It's going faster as you'd expect. But now when he turns off the A current, there's very little effect. In contrast, if he looks at the calcium-activated potassium current here in this network at 10 degrees, and he turns off the calcium-activated K, the network is fine. It's going a little more slowly, but it's fine. But when he does the same thing at 25 degrees, you can see here, turning off that current completely disrupts the network activity. So this gives you, again, a heuristic way of seeing the effects of what we call state dependence. And if you take this seriously and think about pharmacological treatments with individuals with different starting underlying parameters in response to different perturbations, you realize why it can be so difficult to get pharmacology to work predictably across individuals. And I think it's just really very interesting to see it so simply played out. Now, this just summarizes what I've just shown you, that individuals' similar starting circuit performance with relatively similar circuit mechanisms in the control state can nonetheless show entirely different crash mechanisms. And I think this is a cautionary tale for thinking about disease. And then I just want to come back to something I briefly said to you. Randomly sampling parameter space shows that neurons that are temperature compensated are not common in the population. And we had to search really hard to find them. But each crab has to find these neurons automatically, right? So how does it do that? And I think we know a half answer to that. I probably don't have time to show it all to you, but I'll very briefly indicate what the answer is. And we know that the components of functional circuits are not static, but they're constantly turning over. Channels are coming and going in the membrane constantly. And so every single cell in your brain has to rebuild itself all the time. And so there have to be biological mechanisms that allow cells to do that. Many years ago, first with Larry Abbott and then later by ourselves, we built a series of homeostatic models, which basically said that the activity pattern was the controlled variable and that cells could use a target activity level as a way of rebuilding themselves to a target activity. So in Tim O'Leary, when he was a postdoc in the lab, some of you probably know him, built a new homeostatic model. And in this model, we have intracellular calcium concentration, controlling the rates of turnover for each single ion channel separately. These models are simple negative feedback where there's a target calcium concentration, and the models actually change the synthesis and degradation of the ion channels in the membrane when the intracellular calcium deviates from the target. Well, if you do that and you start with random initial conditions, you can build cells. And then interestingly, these rules, if you run the model over and over again from different initial conditions, and these are 6 different models, here you are seeing 6 different models with different final conductance densities. Here we've recovered that 2 to 6-fold range. Again, this is on a very compressed time scale. Even though their behavior looks very similar, they have different underlying structures. But the cool thing is, and this is why I showed you the correlations, if you look, you discover that these homeostatic rules develop correlations in the expression of these ion channel genes. And if you start with those correlations, then Tim was able to find pretty easily some models that were all wonderfully temperature compensated. So to summarize, what this means is that we think that biology creates some rules for the correlated expression of ion channels, and that's part of determining a cell's identity. And when those correlations in channel expression are put in there, then you're on a manifold that allows you to be, for example, well, temperature compensated. You don't find these solutions in random parameter space, but you basically have to be in the right part of correlated space. And so we think that over evolutionary time, these correlations have been found, which is why every single crab that we've ever studied does perfectly well from 7 degrees to 25 degrees or whatever it is, and only starts showing the bad properties as we go higher. So correlations arise automatically in these self-tuning homeostatic models. These models generate variability in multiple cell types. I didn't have time to show you all of this. And I didn't show you the rest of the pathology, but I'll just end by saying that differential resilience in the population is a necessary feature that comes from the inherent degeneracy or multiple solutions producing similar behavior in the healthy nervous systems. Some mechanisms for differential resilience are automatic and arise from the properties of ion channels, receptors, and signaling. And then some mechanisms for resilience will depend on homeostatic mechanisms, behavioral adaptation, and learning. And I'd like to thank you, and I'm sorry I went a couple minutes long.
That's great. Thank you for the wonderful talk. We have time for a few questions, and I'm gonna read some here that have been upvoted and percolated up here in the questions and answers. So the first one is from Paul Sisek, and he asks, when you get similar dynamics in terms of spiking patterns using different settings or parameters, are the parameter sets like behave like a continuous subspace within the total space, or are they scattered clusters, right? So in other words, I think what he's getting at is there noise correlation in the variability of these parameters, right?
Well, first of all, if you look at all the multiple spike bursters, they're actually in a connected region in parameter space.
Mm.
Look in tonic. If you have enough currents, so that's another thing I didn't bring out. But if you only have two channels, sodium channel, potassium channel, then it's very easy to go into many islands. But by the time you have 6 or 8 or 12 different kinds of conductances, there are many paths in the multiple conductance space. So we think that many of them are on these large connected manifolds with that 2 to 6-fold range, giving you the dimensions on which they have to stay. So, for example, in you, your spinal motor neurons are constantly turning over the number of channels. So they're not sitting at a single point. They're wandering around on that manifold throughout your lifetime.
Right.
So I think if that's what he was asking.
Yeah.
I think that's what he was asking.
Very cool. Thank you. So moving on to the next question here, which I think will call for some speculation. Do you think there is some epigenetic programming that allows the parameters to be set to match the environment during development?
Yeah, I'm sure. So because in animals, so for example, Calnectes sapidus, which is the blue crab, it lives in environments from Rhode Island down to the Caribbean. And I'm sure that the animals that grow up at 30 degrees are sitting with a slightly different set of underlying biophysical and molecular properties than the ones that are growing up in Rhode Island. So I think there must be long-term epigenetic changes that put you into one set of correlated spaces. You have the really fast things and then slow things like neuromodulation, and then the very slow things.
Very cool. So, by the way, this was a question by David Damaris. Sorry, I didn't mention this earlier. But that dovetails nicely to another question by Jan. Basically, he asks, what makes all of these different biological circuits actually converge to the same output pattern, right? Like, so what's the pressure to have that stable pattern at 10 degrees Celsius, for example?
Well, I mean, what we're putting in developmentally is LP always makes a synapse with PD. PD always makes a synapse with LP. LP always makes a synapse with PY. PY always makes a synapse with LP. There's always a strong electrical connection between AB and PD. So there are developmental rules that set up the backbone of the connectivity. And once the backbone of the connectivity is there, then the tuning of the conductance densities is done by probably experience and long-term whatever. So it's actually the backbone of the connectivity basically enforces the triphasic rhythm to a certain degree. And the animals that don't develop triphasic rhythm died way before we got them.
So it's natural selection.
We don't know. So we don't know what fraction of animals never made it past 3 months. We're seeing them at age 6 years. So we're only seeing the successful ones.
Interesting. Very cool. All right. So there's another question here from Andre Marquez Smith. Are the responses to perturbations constant at the ion channel level, or every time you perturb the system, does the compensation follow a different trajectory?
Okay, so we can't do that experimentally easily. What we know is, well, I guess we can to some degree. If you take the same neuron in voltage clamp and measure the densities of some of the K channels, and now you perturb it for several hours, you can show that those channel densities change. And then if you wait, you can go back. So to some degree, it's reversible. But I thought the questioner was going to ask what happens at the circuit level if we crash the circuit and then go back down and then crash it again, how constant those are. And we've been trying very hard to figure out how reliable multiple crashes of the same animal are and how different the crashes in different animals are relative to how different the crashes of the same animal. It just turns out to be experimentally very difficult to get those data. But I think we now have a way of characterizing the networks well enough to be able to do that. This was something where I have a postdoc, who is now using machine learning types of methods to characterize the dynamics of these crashed circuits. We didn't have a good way of characterizing the dynamics of the crashed circuits, and now we do. So I think we'll be able to get at that question.
That's fascinating. So I have a final question then to ask. So most of these experiments are done in fairly stable, very well characterized circuits and involve sort of bursting patterns. Do you think similar properties extend to, say, more plastic networks where maybe just single spikes are the norm, or is this something that's more specific to the circuit itself that you're studying?
I'm sure that everything we said today is true of every neuron in any animal.
Very cool.
Remember, you're seeing if you're recording extracellular from the middle of a monkey, you don't know what's going on.
That's true.
You're just seeing when it's spiking. So you just don't know. And remember, those monkey neurons have 20 kinds of channels in them too. And so, there's a whole other level of complexity that we don't see, which is when each neuron in a population fires and how that population activity gives rise to behavior. That's a problem we don't have. That is present in large networks, but I don't believe for a moment that any of the neurons in the human brain are any simpler than our neurons.
Right. Well, on that note, yeah, I think you're absolutely right. So I'd like to thank you once again for the amazing talk, and I'll invite you to stick around for a very nice to listen to a very nice panel session. So in a second here, we'll be transitioning towards, well, not a panel session, sorry, a Q&A session that's going to be hosted by Sarah Sola and students committee here that's running Maine. And so once again, thank you again, Dr. Marder, and everybody else, please stick around for the